Hello everyone, welcome to the class of biotechnology. My name is Dr. Farah Maarfi. In the last video, we were discussing about the restriction enzyme type first that were composed of three subunits: specificity, restriction, endonuclease, and methyltransferases. Altogether, they work as a single unit and makes a cut around 10,000 base pair away from the cleavage site in the presence of ATP, Mg2 plus, and SAM. Today, we will discuss about the restriction enzyme type second. Actually, these enzymes are mediated by two subunits, that is the restriction subunit and modification subunit. Restriction subunit and another is modification subunit. Although they are different, but they recognizes the same target sequence. So they recognizes the same target sequences, and these sequences are the restriction sites. These are actually the restriction sites where particular enzyme comes and make a cut over there, and these are palindromic in nature. In case of restriction enzyme second, these are palindromic in nature. Palindromic in nature, and these palindromic sequences are either interrupted sites or continuous sites. Interrupted or continuous. Example for the interrupted one is BST E second, and for this the restriction site is GGT NACC 3 prime 5 prime. N here represents the any nucleotide. It can be any nucleotide. For the continuous one, the example is KP and first, and for this the restriction site is 5 prime GGTACC 3 prime. Let us understand the working mechanism of the restriction enzyme second, means how it actually makes a cut on a double stranded DNA. Suppose this is a DNA, right? This is a double stranded DNA, and at this point GGATCC, it carries the restriction site for enzyme BAM H1 second. BAM H1, right? And you all know that BAM H1 is an example of restriction enzyme second. So when the enzyme BAM H1 comes in close proximity to the double stranded DNA, then in the presence of Mg2 plus as a cofactor, Mg2 plus here act as a cofactor, then in the presence of Mg2 plus, this BAM H1 binds non-specifically on the double stranded DNA. Non-specifically means it attaches anywhere, right? It attaches anywhere on the double stranded DNA. That means away from the site of the restriction digestion, or the site where it should actually make a cut. So after binding, it follows a hooping mechanism, or you can say jumping or sliding mechanism, and makes a linear diffusion in the direction of the site. Once the target restriction site is located by the linear diffusion mechanism, the recognition process, or you can say the coupling reaction, triggers the large conformational changes that leads to the activation of the catalytic center. So as soon as it catalyzes the reaction, the catalysis results in the hydrolysis of the phosphodiester bond, as here in the example it cuts in between the two G's like GGATCC. So it makes a cut in between these two G at phosphodiester bond. As soon as it makes a cut at a phosphodiester bond in between these two G's, it leads to the fragmentation of the double-stranded DNA, as in the example, fragment 1 and fragment 2. At the same time, the BAMH1 detached itself from the double-stranded DNA and removes out of it. These two fragments carry overhanging regions or flanking regions. The overhanging regions also called as flanking regions. Flanking regions. And these regions are single-stranded DNA, as seen here in example CTAG, and for this fragment CC, sorry, not CC, CTAG. These, in this case, especially in the case of BAMH1, it generates sticky ends. The fragments here are of sticky ends type, and these sticky ends help in the cloning. By using the enzyme ligase, you can ligate this particular double-stranded DNA into any vector for the cloning purpose. Another type is the restriction enzyme third. These enzymes recognizes and methylated the same DNA sequence, but it cleaves around 24 to 26 base pair away. Suppose this is a site where it should make a cut, but instead of making a cut over here, it makes a cut around 24 to 26 base pair away. And these enzymes are having two subunits. One is M subunit and another is R subunit. R subunit. M subunit actually is responsible for the recognition and modification of the DNA sequence. Modification of DNA sequence. And the other subunit R is responsible for the nuclease action. It is responsible for the nuclease action. That is, it is responsible for the hydrolysis of phosphodiester bond. So as soon as the restriction enzyme third binds to the restriction site in the presence of Mg2 plus, ATP and SAM, it catalyzes the reaction of hydrolysis. It catalyzes the reaction of hydrolysis here. Hydrolysis of what? Hydrolysis of the phosphodiester bond that is present in between the two nucleotides. Here initially it cleaves the single-stranded DNA only. It cleaves the single-stranded DNA only. It does not make a cut on both the strands at a time. Example for this type of restriction enzyme is PST second, PST second, 5 prime, CTGATG, and there is ECO P151, and the recognition site is for the ECO P51 151 is 5 prime CAG 3 prime. Another example is ECO P1 first 5 prime AG ACC 3 prime. Next is HIN third, and the recognition site for the HIN third is 5 prime GG AAT 3 prime. These four examples of the restriction enzymes recognizes asymmetric sequence. So these enzymes recognizes the asymmetric sequences. The last one is the restriction enzyme type 4. This type of restriction enzyme recognizes and modified DNA. This is a modified DNA. Modification might be due to the methylation, that is, it might be of methylated, or such types of DNA might be hydroxymethylated, or they might be glycosyl hydroxymethylated. Any type of modified DNA can be treated by using the restriction enzyme type 4. And the example for this type of enzyme is MCR, BC, and MRR. These are the system of E. coli. These are from the E. coli. More than 3,000 of restriction enzymes have been studied till date. 3,000 restriction enzymes are being studied till date, and more than 600 of them are commercially available. So, from the 3,000, around 600 restriction enzymes are commercially available for the molecular cloning purposes. The table here represents the difference between these four types of restriction enzyme, that is type 1st, type 2nd, type 3rd, and type 4th. So for the example of the type 1st is ECO K1, ECO A1. And for the type 2nd, it is ECO R1, ECO R5, KP, and first BAM H1. These are the examples of type 2nd. For the type 3rd example is ECO P151, ECO P1, HIMF 3rd, and for the type 4th is BAC 1st and BAC 1st. When we talk about the cofactor, then Mg2 plus ATP and SAM is required for the catalysis purpose. For the type 2nd restriction enzyme, Mg2 plus is only required. For the type 3rd, Mg2 plus ATP and SAM is required. For the type 4th, Mg2 plus and SAM is required over here. Next is for the recognition site. So if we talk about that, how the particular type recognizes the recognition site, then it recognizes the interrupted one. Interrupted means that if there is a particular restriction site, then there might be possibility that a single nucleotide in between may be of different types. Next is the type 2nd, that recognizes the palindromic sequences only, and it can recognizes both type, that the interrupted one or the continuous one. This we have already discussed before. And the type 3rd is the non-palindromic sequence, that means asymmetric ones. And the type 4th recognizes the interrupted sites. Next is for the cleavage sites. It cleaves type 1st, that is, it cleaves 1,000 base pairs away. Type 2nd at a specific site only, that's why it is widely used in the cloning purposes. The type 3rd, it recognizes 25 base pair away from the recognition site. And the type 4th, it cuts both a strand and leaves the 3' H overhang. Next is the subunit and the structures. So if we talk about its structure, then it is a pentameric type. Pentameric means that it carries two restriction sites, two methylase transferase enzymes subunit, and one is specificity subunit. And these all together works as a single unit. This type of structure, or you can say this type of subunits are called as heterodimers. Next is type 2nd. It work as homodimer. That homodimers means it have a two recognition sites, or you can say the modification site, and one is the specificity, for the specificity. The next is the type 3rd, that is have both the subunits, that is R and M. M is for the modification and the recognition, and R is for the nuclease activity. And the fourth type is the heterodimer type, and it carries two recognition and modification system, and one is the for specificity. The last is the methylase feature. 1M and 1S act as a heterodimer here, and heterotrimer is 2M and 1S. Type second, it carries two separate subunits and act as a single monomeric. Next is the type third, that is methylates adenines. So it cuts only one strand independently. The fourth one is same heterotrimer, that is 2R, M and 1S. R is the recognition, M is the modification, and S is the specificity. So that is all about the restriction enzyme type first, second, third, and fourth. And these all are having wide range of application, especially the restriction enzyme type second is widely used in cloning purposes, protein expression, DNA mapping, RFLP, preparation of the DNA libraries, and studying of epigenetic modifications. We will stop here today. Thank you so much for watching this video. If you like this video and gained some knowledge, please like and subscribe the channel. Don't miss to hit the notification bell so that you cannot miss any future video. Thank you so much.